Hello, everyone. Welcome to our continuing podcast series, Empowering Proteomics and Transcriptomics in Alzheimer's Drug Discovery. Today is episode number 7, From Molecules to Meaningful Practice, emphasizing heterogeneity and context of use in the development of next-generation biomarkers. This episode is coming to you live from Big Sky, Montana. We're at the 2026 Winter Brain Conference on brain research, and we'd like to thank the conference organizers for generously allowing us to hold this podcast as a part of the meeting. We'll include a link to the meeting, and also for more descriptions on those who want to learn more about the Winter Brain Conference.
Yes. Hello, everyone. We are moving from the last episode from sunny San Diego to snowy Big Sky. We would like to thank Frontier in Molecular Mechanism of Neurodegeneration for their continued support of this conversation, along with the International Neurodegenerative Disorder Research Center, the campaign to prevent Alzheimer's disease, and the Brain Watch Coalition. We are grateful to all of them for supporting spaces where longer form, interdisciplinary, and globally discussion like this can happen.
So to put a little bit of context in the environment today, it started at minus 18 degrees Fahrenheit. And so we are very happy and very fortunate to have two guests with us. Professor Aliza Wingo. Aliza, hello. Thank you for joining us.
My pleasure.
She is a professor of psychiatry at the Albert Holmes Rowe Endowed Chair of Genetics at the University of California, Davis. Aliza's work spans genomics, proteomics across neurodegenerative and psychiatric disorders, and she's been instrumental in highlighting the shared molecular risk signals that challenge traditional disease boundaries.
We are also joined by David Crosslin, an associate professor in the Division of Biomedical Informatics and Genomics in the John Deming Department of Medicine at Tulane University School of Medicine in New Orleans, Louisiana. David is also the director of the Biomedical Informatics and Graduate Program in Biomedical Sciences, the BMS. His work focuses on data integration of genomics data into the electronic health record of clinical decision support work that advances the national electronic health information infrastructure in support of personalized medicine. David brings a critical perspective on how biomarker and genomic signal perform once they move beyond discovery into the clinical systems. Welcome, David.
Thank you.
So the first question for both of you, we're very excited to have you for the conversation, and this conversation going to be a kickoff of our panel session this evening at the winter break.
Yeah, so we're really looking forward to using this conversation to explore the sort of the insights in terms of how heterogeneity, real-world evidence come together and sometimes collide in biomarker development.
And the first question is for both of you. Aging is a major factor for Alzheimer and Alzheimer-related disorder, and also the Lewy body spectrum disorder. What is your perspective, whether this group of spectrum of neurodegenerative diseases represent acceleration or dysregulation of aging?
Oh, well, I don't know if I'm going to be able to approach that, but going back to the real world evidence, I do think there is a treasure of data that you could mine and that could be translated into that type of prognosis.
And Aliza, what is your take?
I think that the aging or the time window in life plays an interesting role in what I have seen in the research, that the people who are diagnosed with a psychiatric disorder, which is the brain disorder, in early or midlife have higher risk for developing dementia, Alzheimer's related dementia, and up to 6 times higher. And through our work and others, we have found that supportive of that epidemiologic connection is the genetic connection. There is some shared genetic basis between the psychiatric conditions and late life neurodegenerative diseases. And why does this matter? It matters because psychiatric disorders affect 1 in 4 or 1 in 5 people in the population. So it's relatively common. And so it is interesting that we see some genetic links as well as some shared risk genes or risk proteins. And so I think this opens up a very interesting opportunity for early prevention and precision therapeutic development. So I think age here, the connection is that the psychiatric disorders typically manifest in early or midlife, and neurodegenerative diseases like Alzheimer's disease or related dementia or Parkinson's disease typically manifest in late life.
And let me even further reframe the question and connect it to biomarkers. So you both work on molecular signal that gives window into brain changes earlier in life, like you just mentioned. And Aliza, you mentioned very nicely your work on proteomics as a signal for psychiatric disorders that overlap also with dementia and other neurodegenerative diseases that can explain to us changes in the brain on the proteomic level that happen years before dementia happened. And David, you work on genomics, which we can characterize very early in human life, the genetic variant. So how could that help in reshaping the discovery and the development of the next generation of biomarker for neurodegenerative diseases?
Well, are you talking about genetics or all
For you,
omics?
genetics or
Yeah, I mean, I guess it's a common theme now, but linking electronic health record data to any omics is a powerful tool. So no matter if you're looking at comorbidities that were just mentioned, you can easily translate that into discovery. And so we do what's considered a FEWAS. So no matter if it's ApoE, if it's a polygenic risk score, if it's a proteomic profile, you can go back and look for associations based on diagnosis and procedure codes and visualize that within a FEWAS plot, which is like a reverse GWAS. And so it could easily translate into finding other comorbidities that could cause that.
And we can do it very early in life.
Yeah, absolutely. Anything retrospective, and that's why we have these large data repositories where you can go and assess that.
So David, I'm sorry. I was just going to ask David a follow-up question real quick though, because you spend a lot of time living at this interface between sort of this clinical data and this very technical molecular level signatures. Can you think back when you first entered this space, what did this look like in terms of trying to deal with electronic health records and sort of the totality of their tidiness, said sarcastically?
Yeah, it was, I have a timeline that I'm going to present. And this was the NHGRI, who's probably funded 90% of my research. And their hypotheses was that you could link this for multiple discovery. No one knew what they were doing. and this was 2009. And, I was a postdoc at that point. And, when you're a postdoc, you agree to do anything. So I remember going to the coffee shops on Saturday, just trying to, with secondary use of data, there's a lot of noise. So being able to clean that data for discovery is where we do a lot of work. So to answer your question, no one knew at first, and then all of a sudden the discovery started really pouring in, and it was wildly successful. And we continue to do that. And I use it as training for graduate students now. And so we're developing, the new scientists to really understand and appreciate it.
And Aliza, what is your opinion? from your
About
perspective, the
early,
early
early-
biomarker?
Right. So related to electronic medical records, it's a really great resource because I was thinking that if we find risk proteins or risk genes that are shared between psychiatric conditions that manifest earlier in life and the neurodegenerative diseases that manifest later in life, if there's something shared, that shared risk genes could be targeted to hopefully treat the psychiatric disorders, but can also reduce the risk for dementia. And of course, that's for new targets, but we can also do drug repurposing. If there are some targets that are already, some genes or proteins that are already targeted by existing medications. For example, ACE is a protein, a risk protein in Alzheimer's disease, and it's also a risk protein in depression, and I can't remember in other conditions. So ACE inhibitor is a medication that's prescribed for high blood pressure, and we can use electronic medical records because that can span years, 10, 15 years, to see if people who are prescribed ACE inhibitors earlier in life, maybe in the age window of 50s, early 50s, whether their risk for dementia would go down. I know that has been done, and the results are mixed, but we haven't taken into account the individual genetic risk profile. And that is going to be another layer of information that we can add to the analysis to see if genetic risk factors can influence the effects of a particular medication on the risk of dementia or depression.
Is there of these sort of these shared risk proteins that you're describing, is there one that sort of stands out as particularly interesting or mysterious that, you know, sort of merits deeper insight, and particularly in terms of the idea of looking at drug repurposing?
ACE inhibitor is one that I can think of, and then related to that, calcium channel blockers,
Okay.
even though they're not, they're a little downstream from the direct effect on ACE, but the downstream effect of ACE can be targeted by calcium channel blockers. So those are the two that come immediately to mind. I think there are other medications that, such as metformin, that target, I can't remember the name of the gene, but there are some other candidates that we can look
But
at.
working on that cardiovascular metabolic pathway sort of as a means of intervention.
Right. Yeah.
No? I know that Ara is very excited from you mentioning the calcium channels.
Yeah.
So I will ask you another question. Which role do you think proteogenomics is strongest as today in discovery, patient stratification for subgroup of patient, or prediction of disease?
So proteogenomics, we can look at different tissues. Our group focused on brain proteomic data, but you can also look at CSF proteomic as well as plasma proteomics. We have been working the longest in the in brain proteomics, so I can speak more to that. So we think that if we use proteomic data from the brain, that's the organ where psychiatric disorders or neurodegenerative diseases are, I think, most relevant. So I think brain proteogenomics can help elucidate the biology underlying these conditions and can be instrumental in nominating potential promising genes or proteins that we can perform functional validation and therapeutic development. So I think that the underlying pathophysiology or pathogenesis and drug development is brain proteogenomics is instrumental in advancing that area. In terms of the biomarkers, CSF is the tissue that is the interface between brain and plasma. And of course, plasma is easier to get the sample off. CSF is a bit more challenging. Brain is the most challenging. So I think CSF proteomics can help with early detection or biomarkers for early detection, as well as plasma proteomics.
I want to get David to jump in on this because this is going to be part of our presentation. But in terms of these biomarkers, there's many levels of validation. But where do you see, or where have you seen these somewhat promising biomarkers basically sort of fall apart in the clinical translation from even past clinical research into clinical systems and clinical care?
Well, I mean, the monogenics are already there, whether you're talking about ApoE or other single genes that cause effect. I've been working on a study where we're taking polygenic risk scores and putting those back into the electronic health record and working across 10 conditions and assessing the impact. Now, the question is that one, implementation science. One, does the new diagnosis tool work? Mm-hmm. And the second question is, does it work in a clinical setting? And that's where the implementation science comes in. So you could have the best diagnostic tool, but it could be impossible to put into a clinical system to actually make an impact. So they're instead of looking on a patient level, you start looking at a clinic level, an office level, a health system level, as opposed to a single level. And that's a whole different type of research. It's called dissemination and implementation. It's actually where my research interest lies now. And so no matter, I keep on going, no matter what tool you have, just understanding how you put it into practice. And, there's science about specimen collection within the clinic visit, getting the results back, integrating that into the electronic health record and clinical decision support. And so it's fun, it's hard, it's busy, and yeah.
And from your perspective, so where do you think that is the biggest gap between the molecular signal and the actual functional outcomes? So it's a related question, bringing it from the proteome.
So the goal is, of course, for someone, I'm a clinician, I do see patients, and I see there's a pressing need for more effective treatments for neurodegenerative diseases as well as psychiatric disorders. So early prevention is always great, but then the treatment is another area that I would like to see new treatments that are available. And I see the gap between what we have found. We have built on findings of genome-wide association studies that's 10 plus years in the making. We added to those findings human brain omics data, transcriptomic, microRNAs, proteomic data. And so we combine the information from GWAS and human brain omics data. Now we come up with a list of risk genes or risk proteins. The next step would be to validate these functional validation of these before we can see which one we can develop therapeutic medications or treatments for those. And I think the functional validation is the next step. And for neurodegenerative diseases, they are model systems. And for psychiatric disorders or another set of symptoms, behavioral and psychological symptoms of dementia, which is just as challenging for patients who have Alzheimer's dementia or related dementia, and caregivers are not studied as much. I think it's very hard to get a model system to validate the functions of the genes, of the proteins that we found in the behavioral and psychological symptoms of dementia. And I think to find an appropriate or a relevant model system to test, to validate the functions of those risk genes is the next challenge.
I very much agree with you about neuropsychiatric and behavior, but I think even we can take it further. We are not even there for the neurodegenerative diseases.
Yeah, that's right.
And there are what are called hybrid trials, where you can assess the diagnostic tools and whether it could be implemented at the same time. So you're measuring both.
Well, I think we're slowly closing in at the end of our time here. We're going to have to go to our conference, actually. But did you have one last question?
I have one last question. Again, it's very related to Aliza. You mentioned the accessibility of plasma versus the inaccessibility of the brain to evaluate a biomarker and to use it for as a biospecimen for biomarker, and then the CSF as between. But let me just throw out there the hypothesis that these neurodegenerative diseases, brain diseases, could be systemic diseases. Mm-hmm. Then it's open a whole new space for evaluating biomarker, maybe even earlier. Yeah. But also in tissues that will be more accessible. So and this is from your perspective and from David's perspective, again, we're talking about genetic variation. We can do it in many tissues that are accessible. So I would like to hear each of your perspective for our closing.
Yeah, no, I see your point. I think the immune system is very powerful, very relevant, and I think it's a part of the biological processes. I envision that there are many biological processes that are involved in neurodegenerative diseases, not just from the brain or from the immune system. I think it's probably a confluence of the different systems. So yes, the immune system is an important component, but I think the biological processes in the brain are just as important.
And David?
Yeah, I feel like we're just scratching the surface. And I mean, we still don't know what DNA is doing all over and then you start going further downstream with what you're talking about. I think you just, you start with DNA, start working your way down, integrating, and, I think it'll all come together eventually, and I look forward to it.
So my last question is, fast forward 5 years, what do you think, or what would you hope that we'll be doing differently in terms of biomarker implementation that we aren't normally getting done right now? Where do you see the innovations coming in terms of the implementation?
Well, I personally would like to see biospecimen collection at an earlier age, where you start building these these clinical data warehouses that are linked to biobanks, so you can start to assess this not until it already happens, and then you're going and look at that. So I would say the technology to really integrate this, and to get the payers involved and the insurance companies to buy into this, I think would be where I would like to see it go.
Yeah, great point. And I would like to see that it's easier to get access to existing omics data and electronic medical records. Right now, there are still challenges with that. And also, I like to be able to see that multiple genes are targeted in a pathway instead of just one gene or two genes, because I think these are polygenic conditions, and a lot of genes act together to cause the disease, and we haven't had a way to target multiple risk genes together.
I'm sure this is a common space between both of you, the multigenetics factor. Yes. So I would like to thank you both for joining us today. I'm looking forward to extend this conversation in our winter brain session this evening. And to our listener, stay tuned for episode 8, where we will continue the journey toward precision medicine by going under the woods of integrating genomics and patient care.
Until then, I'm Ara Khachaturian with Ornit Chiba-Falek. Thank you for joining us, and we'll look forward to seeing you very soon.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you for having us.